In this presentation, we're going to be talking about the production of energy in a cell, which is also known as ATP. The metabolic pathway that we're going to be focusing on is the electron transport chain, which takes place in the inner mitochondrial membrane and also the matrix. These symbols are going to be used in the presentation to represent the following molecules that are involved in the production of ATP. The ETC is composed of 3 transmembrane proteins and 2 transport proteins. The first transmembrane protein is called NADH hydrogenase. The second one is cytochrome BC1 complex, and the third is the cytochrome oxidase complex. The 2 transport proteins are Q and cytochrome C. These protein components of the ETC are arranged in order of increasing electronegativity, with the weakest attractor of electrons, NADH hydrogenase, at the beginning of the chain, and the strongest cytochrome oxidase at the end. The ETC is also composed of ATP synthase, which is the final protein that synthesizes ADP in an organic phosphate to produce ATP.
Initially, there is low concentration of hydrogen in the inner membrane space and high concentration of hydrogen in the matrix of mitochondria. The goal is to transfer the hydrogen from the matrix into the inner membrane space. Eventually, the NADH travels to the NADH dehydrogenase and gets oxidized. It then gives its 2 electrons to the first protein complex and 1 hydrogen to the matrix. The 2 electrons then pull the 2 hydrogen from the matrix into the inner membrane space. The electrons then get transported from the NADH dehydrogenase to the cytochrome C complex by the Q component. Cytochrome C pulls the electrons one at a time from the cytochrome BC1 complex to the cytochrome oxidase complex. As the electrons travel from the cytochrome BC1 complex, 2 more hydrogens are pumped from the matrix into the inner membrane space.
The next molecule that provides energy to produce ATP is FADH2. FADH2 travels to the transport protein Q, gets oxidized and gives off 2 electrons to the protein complex and 2 hydrogens to the matrix. The electron then travels from Q to the cytochrome BC complex. The potential cytochrome C pulls the electrons from the cytochrome BC complex one at a time to the cytochrome oxidase complex. As the electrons travel to the cytochrome oxidase complex, 2 hydrogens are pumped into the inner membrane space.
By the time the 2 electrons from NADH and 2 electrons from FADH2 reach the cytochrome oxidase complex, they are in a very stable position. The oxygen in the matrix, together with 8 hydrogens, reacts with the 4 electrons to produce water.
As the electrons travel into the inner membrane space, the concentration of hydrogen increases in the inner membrane space. The high concentration of hydrogen creates an electrochemical gradient. The energy in the gradient produces a proton motive force, which pushes the hydrogen toward the ATP synthase. The potential energy released by the hydrogens as they move through the synthase causes a change in shape. This change in shape opens up the active site of the ATP synthase and joins an organic phosphate in an ATP molecule.
The final product of ETC from joining an inorganic phosphate and an ADP molecule is an ATP, which can then be used by the cell as an energy source.